appreciate that we become so appreciative as we have worked from home for 6 months now.
I see.
And it's it's imagine that get into the subway on a Monday morning and then be physically reporting to the site every day, 8:15 in the morning, Monday morning.
Yeah, we still we still do that.
With my heroes.
So whenever you're ready, we can start. I already started recording.
Normally, when would you like to start? It's
Yeah. Usually we start at 8:15. So most of the people are probably attending remotely today because there's a lot of people on Zoom.
okay.
So yeah.
All right. Okay, great. So let's get started. Hi. Good morning, everyone. And as I said to Dr. Alloway, um, pleasure to present to you the second part of the COVID-19 test series. Uh, as I said, I appreciate we as a general population really appreciate for all the heroic efforts that you guys have been doing and put your life in the upfront to make sure that the society is still functioning and make sure that everyone is taken care of. So it is my pleasure to present uh, my knowledge to you, and then but really want to thank you for that. Okay, so today is the second part of the COVID-19 epidemiology series. Um, Today I'm going to focus on the immunity, meaning that first part will be the antibody test, and then second part is what is the current status of the vaccine development. So again, overview of these lecture series. It'll be 3 lectures total. Last week, I talked about the COVID diagnostic test. So the followed by this week, which is the antibody test and vaccine development. And then next week, I'm going to talk about the mass test at scale so that how to get out of the crisis. So first, a bit of introduction. Who am I? Um, I'm a statistician working for Downstate, but I also held a medical degree from China many years ago. In the recent years, since I joined Downstate, I have been working on the EMR data extraction and help us physicians working on their various projects. Personally, I've been very interested in the COVID-related topics. First, I work for the infectious disease, and as you know, that as Dr. Fauci, nobody had a prior experience in COVID before, so all the prior experience medically came from HIV, where I work. And also, I have, as a statistician, I have been interested in working on my own as a hobby, uh, getting the open data source from New York State. So I have developed a few dashboard interactively on the web. Hi, can you hear me okay?
Yes, we can hear you. Yeah.
Because my screen freezes, that's fine. And then the screen's moving?
Yeah, the it's moving. Yeah, we can see the mouse.
I don't know if it stopped. Um, so a bit background of where we are. Since March, it's been 6 months, so feels like eternity. But the good news is that taken from this September 13th snapshot, the good news is that New York City is doing relatively very well considering where we came from, which we were on the top. So if you look at the left panel, which is the morbidity and mortality in selective cities, New York City is very down below in both cases and deaths per cases. And we're doing pretty solid on the scale of the test. So we're doing fine. And in terms of locally, which is on the right two panels, here is the status in downstate. As you can see that we have very low patients currently in the hospital and in ICU. And in terms of Brooklyn in general, the most cases are in Maimonides and NYU, Brooklyn side. And then the hospitals where you guys work with downstate and then city Kings County, relatively very few cases. Can you see my mouse moving? I'm sorry, Halloway.
Yes, I can see it moving.
Thank you. Perfect. Thank you so much. Okay. So, but still, let's do not forget that even that we're doing relatively fine. There is a slight chance of uptick. So still be careful. And state-wise, the indoor dining is still not open. And then Governor Cuomo has declared disaster through October the 4th. And then there has been travel advisory from 29 states. So even that the infection rate has been below 1%, currently at 0.8% for the past 30 days, we cannot take too easy. So recap of last week's when we talked about the diagnostics text, I focused on the relative importance of sensitivity, test frequency, and turnaround time. Generally speaking, for any test, sensitivity is important, but in this COVID pandemic situation, the testing frequency and turnaround time should weigh more than the sensitivity. So it is very important to test general population as frequent as possible and get a turnaround time as quick as possible, meaning within 2 days. But on the other hand, there in the hospital settings, which is especially high risk settings, the sensitivity is still relatively important for your protection and for you to prioritize patients' flow. In terms of transmissibility, again, here is the timeline. In terms of viral load, the peak infections starts before the symptom onset from -1 to -3 days, and then it peaks 1 week after symptom onset. And then following the viral load, the antibodies start to develop around the first week, and then it peaks around the 2 to 3 weeks. And I'm going to talk about more in general. And then, so that affects, these timeline affects the testing sensibility. The red line, which is the PCR diagnostic sensibility, the orange line declines over time as viral load declines, and then concurrently, the testing, antibody test sensitivity start to rise and then stays to 100% sensitivity. So that is due to the time frame of days since onset. And I want to finish up last week's diagnostic test, which I didn't go over in the interest of time. There has been some debating areas on diagnostic tests. One is that, what about the false negative, infectivity, and quarantine? So if a patient is testing negative, meaning he or she is truly positive, should we worry about their infectivity? Um, the answer is not so much. We can assume that they are not that infectious anyway. And then what about the current 14-day standard quarantine time? Well, there has been a growing consensus that due to the real incubation time, which is 1 week, the quarantine time of 14 days is kind of a overkill. So people are leaning more and more towards 7-day quarantine time, just to let you know. Then what about the symptom severity? Is the symptom severity associated with the viral load? And do patients who have a higher, a more severe symptom have a higher viral load? The question's not really. Symptom severity may or may not be associated with a higher viral load. It's more associated with the patient's own immune response, for example, and the time frame. For example, when the patient is on the intubation, by the time he gets on there, his viral load is so low that it's almost undetectable, which is in a way good news for front and the worker, like physicians' protection. Remember that the peak infectivity occurs before or right after the symptom onset. So, and then second, what about the specimen collection? How to evaluate the newly approved patient own collection at home? The answer is that because the instruction is clear enough and then the storage mechanism is good enough, there is actually, as you can see from this right side panel, that there is a high correlation between patient's own collection versus healthcare workers' collection of tissues. So patient collection is pretty good. So which is also good news for the healthcare workers that will decrease the exposure for you guys in terms of sample collection. So moving on to outline of today's topic. Today I'm going to focus on the immunity. First part is the antibody test. I'm going to talk about patients' own immune response to COVID in specific and how to evaluate the various of antibody tests available. And I want to bring this topic of herd immunity on COVID. You guys have heard about it over and over again. And also some residents and physicians, faculties have reminded me that relative sensitivity, specificity is, and the related curve, ROC curve, area under curve, all these topics are on your board exam. So I'm going to squeeze in to take this opportunity to talk about some theories, which is relevant to that. And then the second part of today's topic is going to focus on the vaccination. So what is the current status of the vaccination? And finally, what about the priority of the population to be vaccinated once it is developed? So we touched about this topic yesterday, the EUA, which is the Emergency Authorization by FDA. So far, there has been 193 tests overall that have been authorized under emergency use, and the majority of them, or 90% of them, which is 158 of them, are antigen related or diagnostic tests, which I'm not going to allude too much further. We talked about the sensitivity and the practicability of these tests. So among them, there's only 33 antibody tests that has been available on the market that is being authorized by FDA. And the antibody tests, generally speaking, they have great sensitivity and specificity, all above 80%, satisfying. So when do our body respond to COVID, to COVID antigen? So the consensus is that the IgM antibodies start to develop from day 0 and then peaked at day 7 and continued through day 28. And then subsequently, a week after that, the IgG antibodies start to develop and then peaked at 28 days and then start to dwindle. So safe to say that 7 day is the peak of IgM antibody and 20-something day is the peak of IgG antibody. And then so the tests are related to that. And then as this right-hand side figure shows that there are various of tests available. Some focus on IgM, some focus on IgG, and some focus on total antibody. So looks like that total antibody, of course, is better than individual ones, but they all have merit on the market. And then in terms of true and false positive COVID antibody test, there is a window, due to the window of these antibodies' existence. So if you're not catching up, particularly the acute antibody, then you can fall into these false negative period. So it's not that the patient has not developed, it is that he has not developed yet, or the acute IgM has dwindled. And so depending on when the patient has developed antibody and when the test has caught the development. And then also on the other hand, the COVID serology is a little different. It has some uniqueness, meaning that it follows generally some features of the general virology, but sometimes it does not follow. So on the left-hand side, we have talked about these. So as I said, it really depends on patient's reactivity. And then in specific to the COVID serology, there is this notion on neutralizing antibody versus binding antibody, which is on the right side panel here. A neutralizing antibody is an antibody that defends the cell from an infection by neutralizing its biological effects. So most of the COVID antibody tests looking at binding antibodies only, not necessarily measuring this neutralizing antibody, NAB. So keep that in mind. Even that NAB, which is the neutralizing antibody, is more desirable, but so far, most of the tests has not been able to catch up this. And then the most closely related to the COVID is SARS. The experience of SARS shows that NAB can remain active for 2 to 6 years. And then the memory cells of B cells and T cells is much longer. So these are important for COVID as well. And then here, these figures shows that on the left side is the body's immune response to COVID, and then on the right side shows that the approved antibody test on the market. So the body's response to COVID, due to the nature that it's mRNA, it's quite complicated. It involves T cell, B cell, and it triggers each other. And then so far, most of the approved tests uses interferon gamma release assay as the test, and then uses this notion as surrogate marker, which is the T cell or CD4. Surrogate marker, the definition of surrogate marker is that when the true marker or the endpoint, which is the true COVID antibody is not available, we measure the total CD4 or T cell as a surrogate for the immune response. So that is the statistical definition of surrogate marker. So just like HIV and SARS, CD4 is generally the surrogate marker, and occasionally CD8 as well. And in terms of the various of tests available on the market, measuring the gamma T, generally speaking, they have a great sensibility and specificity. As you can see that the sensitivity is 91% and specificity is 93% as median. And using T cell CD4 as surrogate marker, that we can see that whether the antibody is measured by is responding to the peptide only or the total virus as well, it both has been pretty satisfying. And this table shows the 9 available antibody tests on the market on the bottom part. And as you can see that it has pretty good results, and the positive rate is very high, and then the negative positive rate is very low. So generally speaking, it is pretty desirable. And the point of care test is has been used as a standard to evaluate, just FYI. And again, these evaluation of 9 commercially available SARS-CoV antibody tests shows that wherever it comes from, whichever lab, whichever company, they has been pretty consistent in CD4 response, as shown on the upper part here, and then bottom part showing the ROC curve, which I'm going to allude further. Just FYI, the more this curve is shifting to the left, or the more space is taking under this curve, the better the test it is. And then this 50% ratio is random, meaning that the test is useless. So all these tests are pretty well, as you can see, that are have a good sensitivity and a good evaluation. Not surprisingly, the total antibody test weighs better than IgA, which further weighs better than the IgG. But overall, it is pretty satisfying. And in terms of the evaluation on sensitivity to duration of illness, this bar graph shows that 2 weeks after the onset, or 2 to 4 weeks, or greater than 3 weeks of these various of 9 tests. Because the antibody is time dependent, the diagnostic of COVID-19 test is also limited to patients depending on which antibody are you testing, and then how well and how soon does the patient develop antibody. So generally speaking, within 7 days of symptom onset, the detection of SARS-CoV-2, the nucleic acid detection is more superior, which is in this bar here. However, as the second group shows here, after 8 days of illness, the sensitivity of serological tests surpasses of that of nuclear test. And then further, as the days goes by, 10 days after the onset, all of these tests are very good and have 100% of serology positivity. So therefore, most of the tests, when they are evaluated, the gold standard is that after 28 days or 3 to 4 weeks. And then the ELISA test and the point of care test, they are very agreeable with each other. Mostly the agreement is between 90 to 95%, with the IgG test as the exception of well below 80%. But still, overall, these are very good agreements. So meaning that ELISA test can be used as a substitute of point of care test due to its practicability. On the other hand, are the immunity equivalent to the antibody-mediated protection? That answer is more complicated than the test positivity itself. In order to define antibody-mediated protection, we have to say that we need a more neutralizing test antibody, which at this moment is not very feasible, where most of the tests here are more binding tests. So just keep that in mind. I want to talk about the herd immunity. Herd immunity, so these are the computer simulated tests that on the left side, which is about half of the people after half of the people are infected, these herd immunity are reached around 20 days to 40 days or up to 80 days, which we are at this moment. Versus if it's on the right hand side, if the 6.9% of people are infected, then it takes much longer to reach the herd immunity. So in conclusion, the true herd immunity threshold probably lies between 7 to 24%. And as you know that the surveillance in New York City, especially in Brooklyn, shows that we are at 25% level. So in a way that the herd immunity is reached in New York City, which is unwittingly quicker than what we realized. And Brooklyn are one of the hardest hit boroughs. So the infectivity, including the silent immunity, has been reached between 25, almost 50%. So the good news is the herd immunity is reaching, even that with or without vaccines availability. And then the other sides of the world that is reaching herd immunity wittingly include Sweden and Italy, which I'm not going to talk much about further. But the question of reinfection and recovery or immunity. We talk about immunity and recovery early on. So immunity recovery may or may not mean immunity. On the other hand, this reinfection rate kind of has been on people's nerve, because there has been a few cases reported in Hong Kong and in other places of the world that suspecting reinfection. but the consensus is that reinfection is very rare, and it's actually suspected different viral genome sequencing. So it is not technically reinfection of the same virus. So we can rest assured. And there has been suspicion that it is the continuous viral RNA shedding rather than the new infection. And remember that immunity is antibody and cell mediated. So all these memory T cells and memory B cells has played in the role as serology, so which may cloud the serology test. So in the consensus is that the reinfection is not really a common thing, and immunity is likely. So if people are recovered, so if that is great news for the patients and for healthcare workers who has been constantly exposed at a low dosage. And speaking of the pathology board exams, on your board exams, there has been, which is showing here, you guys are going to be tested on the distribution of mean standard error, deviation, analytical accuracy and prediction, sensitivity, specificity, predictive value, and the diagnostic ROC curve, etc. So first, what is sensitivity? Sensitivity identifies those people who are having the disease. It is good for ruling out disease when negative. Conversely, specificity is the proportion of person who test negative among non-affected individuals. So the specificity of a test and the ability of a test is to identify currently non-affected individuals. So in specific, in the COVID case, sensitivity is a lot more important than specificity. Our goal is to catch the people who are having a disease rather than are we catching some other disease such as flu. So sensitivity is in a way more important. On the other hand, as I said before, to get out of the pandemic, turnaround time is more important than sensitivity, which is another issue. And then what influences the sensitivity depends on the sensitivity, is dependent on the characteristic of the affected person. And the characteristic, it's not influenced by the characteristics of a non-affected person. And it is not affected by prevalence of the disease. So conclusion, sensitivity is an intrinsic characteristics of the test itself. And in ideal cases, on the left side, you have the sick people. On the right side, you have the well people who are not sick and they don't overlap. But in the reality, there are overlaps, meaning that test is not perfect. There's never a perfect test. So by having some arbitrary cutoffs, we are including these areas of false negative, false positive, and true negative, and true positive. So it is a trade-off that we have to balance. And speaking of predictive values, the predictive value of a positive test is, here's the formula. It is only valid for the sample of the specimens test. And conversely, the predictive value of a negative test, it is only valid for the sample of specimens it tested. So what is ROC curve? ROC curve is curving. It is a curve that is evaluating the overall characteristics of a test, taking into consideration the specificity, which is on the right-hand side, 1 minus specificity, and the sensitivity in general. This 50% straight line, meaning that the test is useless. So the more the curve is shifting on the left, or the more areas, which is highlighted by this pink area under curve, then the better this test is. So judging from the lower panel here, this orange test performs better than this green line test, which further performs better than this dark red, so on and so forth. So that it is. And it takes in the ROC curve takes into consideration of the patients who is without disease and versus the patients who is with the disease, and it's a moving target. So this ROC curves just make curves all these moving two curves together, just in a nutshell how they were generated. So the second part is the vaccination. What is currently in the pipeline in terms of COVID vaccination? This New York Times has been tracing the COVID vaccination. So far, there has been a lot of vaccine in the development worldwide, meaning 27 at phase 1, 15 at phase 2, 9 at phase 3, none approved so far yet. Of course, some countries have even tried to utilize the non-approved test, but that's another story. And then in terms of a clinical trial, here are the COVID vaccine in the US that is currently in clinical trials. As we can see that the leading companies are Moderna, Oxford, AstraZeneca, and Casino. Pfizer, they have been targeting different parts of the virus. Some are RNA, some are spikes, which is the partial protein, and some are full and some are partial. And then there has been three vaccination that is on phase 3, and the projected expectation time is summer next year or the year after. So again, these are the most important ones, which is currently in the phase 3. Judging from the clinicaltrial.gov, 3 are in the final phase, and then the targeted ending is next year summer. But even after let's say even that after they are approved, and there has been some practical issue, meaning that both of them, the Pfizer one and the Moderna one, has to be stored in -70 freezer, which is very hard. Ice itself will not work. So AstraZeneca one is better because it can be stored in 2 to 8 degrees. And then all of them will have to have 2 dosages. So let's say if we have 1 million vaccinations available, that means 500,000 of the people can be vaccinated because each person needs 2 dosages in 3 weeks apart. So where we are now, we are, as this USA Today says, that we are over halfway here. Even that it still seems dark, like in comparison, 7 o'clock in the morning, even that it's dark out there, but 8 o'clock or 9 o'clock is pretty close. So I just want to let you know that since the start of the pandemic, which is 6 months ago, we are halfway here, and then the lights are coming out very quickly. Actually, I want to tell you that the lights are coming out with or without vaccination because our social distancing, the way we measure masks, and the severity of the disease itself, as well as the herd immunity. So with or without vaccine, we are halfway, way halfway. So, so let's say by next summer, the vaccine is developed, fully developed, but it will not be enough for the first wave for everybody. So what are the priority populations to get vaccinated? CDC just issued guidelines on priority populations. Of course, healthcare workers, heroes, you guys are among the first. And then, but remember that other essential workers, people who prepare food, and people who are long-term care facility, and people in the jail, and Navy. There has been outbreaks on the Navy. So it will be administered first to priority populations and then to general population. So, and on the other hand, what if it does not, the vaccination does not meet current target? We all know that the AstraZeneca trial has been on hold since 1 week ago. And then of course the safety, FDA safety board will meet shortly to discuss about. By the way, there's nothing too nervous about this holding situation. It's not rare at all that the clinical trials are put on hold. It is not uncommon. But still, what about our, due to all these unknowns, what about scenarios? So if it happens on time, if it doesn't happen on time, so CDC issued 2 scenarios. But on the other hand, there's still a whole lot of confusion out there due to the complexity of the disease itself and due to all the unknowns and no prior experience. But as I said, I just want to take Bill Gates, who is leading the global COVID vaccination development. He said, one way or the other, we will be out of pandemic at end of next year, with or without vaccination. Why? Because, as I said, yes, it is great that we can have the vaccination and at least administered to some people, but on the other hand, herd immunity works, and social distancing work, and wearing mask work. There has been strong evidence that wearing mask, even that people may still catch disease, but it will lead to much less severe disease situation than not wearing a mask, and herd immunity. So finally, I want to, I really cannot say enough to thank you for your heroic work in the past, and continue to thank you for your continued heroic works for the future. And before we get out of this pandemic, you guys have been work non-stop. So all of you have put your life and in it, and put your life at risk to some extent, and put your loved ones at risk. So we really, really thank you for everything to all the healthcare workers. So thank you. And that concludes my talk today. I'll be happy to entertain any questions. All right, thank you.
Thank you very much, Dr. Han. That was a great talk.
Thank you.
Yeah, so I guess there's no questions. So we'll end it here, and I'll be sending you the recording right away.
Oh, thank you. That's very nice.
Thank you very much.
Thank you.
All right, thank you. And then
Thank you.
thank you for attending. And then as I said, we all work from home, and it is such a, in a way, distant memory for me that be on the subway and then report to work 7:30 on Monday morning. And then so really, really thank you for everything that you have been doing, and then for all the future works. Okay. And
Thank
it's
you
my
very much.
to attend today and then help you a little bit. All right. Thank you.
Thank you. All right. Have a good day.
Have the final session of the topic. By the way, what I was planning on the mass testing, but I'm open to any suggestions on what you guys would like to hear.
Mass testing is a very good topic.
Yeah, that sounds good.
Yeah. We would like to hear about that. Like,
Right. Okay. In the meanwhile, if you have some thoughts on what you would like to hear next week, and then feel free to let Dr. Eloise know, and then I'll be happy to entertain.
sure. Thank you very much. All right. See you next time. Thank you very much. All right. Bye-bye.
So much. Take care. Bye-bye.